Log on, tune in, find out. Another good idea from Cambridge.
Most of you will already know Andreas, of course, from the programme, but I decided I should have another look at the paper that we used, obviously, in nominating for the Rothschild Visiting Professor. It's very impressive. Yeah? A really distinguished international career: Berlin, Tübingen, Kiel, IAS, Princeton. And all of that was actually before he became a full professor at Bielefeld at the age of 31.
30.
30? Oh, well, it's rounding.
But in the last...
Theoretical physicists, I have to subtract two integers. And
Yeah. But I got the document in August 2 in Vancouver, and in August 26 I turned 31.
There you go. Well, that's most
So you're not that far off from the truth.
impressive. And was at Bielefeld up until, I guess, 2003, but been at lots of institutes since then, and joins us from the Chinese Academy of Sciences in Shanghai, which is great. More than 270 publications, incredibly prolific career. So Andreas is talking today about phylogenetic nets, examples and theoretical results. The only other thing I have to say, Andreas, you haven't seen this highly accurate timepiece here. It is an
I'm very happy with that.
Very good. Thank you very much, Andreas.
Yeah, thank you very much for your very kind introduction. I'm very happy to be here and enjoy very much being part of this conference. I have been already also at the Newton before, but only basically for an afternoon somehow on the way from X to Y, but then we met and we all went over with David Penny to Oxford to meet Peter Robinson then. So this was really quite exciting. Okay, but now I'm here for much more time, much more relaxed fashion, so that's really very enjoyable. Yeah, and about time, you see it's examples and theoretical results. I think the examples, when you see them, will be interesting enough, so they may keep up all our time. Then there will be no theoretical results. If there is time left, I will indicate a little bit what the theoretical results are about these phylogenetic nets. So that's a phylogenetic tree. Of course, phylogenetic trees are special examples of phylogenetic nets. And this phylogenetic tree was drawn. He was a very good artist in this way by Ernst Haeckel in 1866. Just 7 years after Darwin's book appeared on the origin of species. It is one of 20 tables that you find in this 2-volume book on Allgemeine Morphologie, general morphology. And Haeckel was really, for Darwinism, what Mohammed was for Islam in the Arab countries. He preached it and fought for it and so on. And so he constructed all these phylogenetic trees. Here, this is of mammals, of all animals. Here are the animals, here are the protista, here are the plants. There is something, of course, we would think today is wrong because the fungi are somewhere on this side, and so they should rather be on this side, as we know now, of course. But how did he construct these trees? Maybe I'll show you the second one I have as well. You don't be afraid. These are all I show, not all 20s. So this is the mammals. Here are us, Homo sapiens. You can barely read it, but maybe the carnivores are here, which is not quite correct. They should rather here be with the Archaeo and Perissodactyla, the cows and horses, somehow are one family. What is amazingly correct, I was quite amazed when I saw it, but everybody knows, of course, are that the Cetacea, the whales, are closer relatives to the cows than both are to the horses. And in between come the hippopotamus. So the evolution of Cetacea, or whales, happened exactly as we would say in Germany, Klein Moritz would imagine it, as any little boy can imagine it. Because there were cow-like beings in Africa, and they saw that there are some food to be had in rivers, so they went into the rivers and went deeper into the rivers and became hippopotami. And then there was even more food to be had, and they went deeper into the rivers, even down the rivers into the oceans, and decided to become whales. That's exactly how whales came about. So how did they know at that time? Of course, from just morphology, basically. And Haeckel did not study all these many species to come up with their phylogenetic tree. There was a classification system that the people had been developing ever since John Ray in England taught in the late 17th century. He was a Catholic, so sometimes he had a good time, sometimes he had a very bad time in those years. Told people that species are invariant. Species don't change. The offspring of a frog is a frog, and the offspring of a toad is a toad, and there's no intermingling between this. This was, of course, the basis for any scientific approach to biology. And that's why people cling to this idea. Species don't change, ever, never ever. What make Linnaeus and other people who came up with this classification scheme of animals and plants and so on were so much against this idea of Darwinism, of evolution, because it was sort of basically endangering the scientific status of biology if suddenly species can change into each other. And it needed just the overwhelming evidence and then the brilliant idea of Darwin on explaining how it could have happened. I mean, Lamarck had claimed the same thing, but his mechanisms were not very convincing. And Darwin's mechanisms were much more convincing, so then it changed. But so what Linnaeus had done, there is this little sentence. Philip came over for green soup. That helps you to remember the Linnaean system, because first there are kingdoms, then there are phyla, then there are, came over, yes, that's the C's, classes, then there are orders, then there are families, then everything is in the genus, so I think the correct plural is genera, and then there are species. So the biologists, long before Darwin, tried to classify all living beings in these 7 ways. And I always mixed it up, what is what and in which order do they come, until I heard this sentence from Alberto Apostolico, Italian scientist, bioinformatician, and so on. And so you can remember this. Of course, it looks like there are just 7 stages. Doesn't quite fit. Sometimes there are more, sometimes there are less, but it still makes sense. Anyway, all living beings, or at least species, had been classified according to the scheme by John Ray, this Catholic guy, who came up with the invariance of species as the basis of scientific approach to biology. And then there were soon other people following him in France, in Germany. Sibylle Maria Merian, for instance, from the Netherlands and Germany, related to the Merians who made all these town sketches, her father or uncle or both. She went over to South America and began studying caterpillars and realized that out of a certain caterpillar, only the same kind of butterfly will emerge, and this will eat only a very specific plant. And then they will produce more caterpillars of that kind that will eat the same plant, and so on. So this metamorphosis of insects wasn't really well established around 1700. And she did that, and she produced marvelous color plates in her books that are, of course, nobody can pay for that anymore, not even Bill Gates. They're in secure hands in some museums or libraries, all depicted there. Always a caterpillar, a butterfly, or beetle, and the plant which the caterpillar would eat. So all these people lived on this idea that organisms are well ordered. They come in this order, according to Kim Philip. And so John Ray actually said that he developed what he called theologia naturalis, natural theology. He said by trying to understand the order that we find in God's creation, we come closer to God. So that's theology. That's why so many clergymen in England, they were happy, whether Catholic or not, with this general idea. So if they did not have to preach, they were perfectly legitimate. It was perfectly legitimate for them to go out and study forms of life and other things, because that was studying God's creation, the order that God had chosen for this world. So it was some kind of theology. That's why you find so many interesting observations by English clergymen in the 18th and 19th century. It all goes back to John Ray. And if you go to Google and now look for web pages of this strange guy, John Ray, of course, there are many web pages, all by creationists. They are terribly happy with him. But after all this was set up, and then with Darwin's theory in mind, Ernst Haeckel just had to sort of give this hierarchical ordering a dynamical interpretation. So, and that's how he came up with all these trees without much research. He just sort of copied what the other people, the taxonomists, had done and put it into tree-like representation. So this would be one kingdom. Everything up here is a kingdom. Everything up here is a phylum. Everything up there then is a class, order, and so on. All what is up further up the branches is either this or this or this. So you easily, once you have the tree, so now we think, okay, there was some first animal, and then there were three different kinds, the animals, the protista, and the plants that evolved from that. And then from the animals, there evolved the Coelenterata, the Echinodermata, and the humans. Actually, also this we know now much better. The Echinodermata are the close relatives of the vertebrates in the world. Much closer relatives of ours than the insects, for instance. But this was also known based already on morphological studies. So this has to be revised and revised, but in principle we all believe that there are such trees. But it's not so easy to construct them so that everybody is convinced. I told you already there's a number of mistakes in here, but how can we really know that what we believe now to be true is really true? So Darwin says, as we have no record of the lines of descent, the pedigree can be discovered only by observing the degrees of resemblance between the beings which are to be classed. Okay, yes. So we look at a horse, at a cow, and at a donkey, and we think the donkey is more similar to the horse than to the cow, so we put it there. Or we look at the dolphin, at a tuna fish, and at a man, and we think the dolphin is more similar to the tuna fish than to the man, so we put it there. Careful, you see, if you look more closely, if you cut us all up, you see the tuna fish actually is further away from the dolphin. So there are all these, how do you observe these degrees of resemblance? How do you measure them and how do you come up with them? That's a real problem. That's the art from which you then, once you have this, you can then define distances between any two species, because that's a mathematical fact. These trees are absolutely determined up to sort of the drawing by the distance between any two leaves. If I have, so the distance of course is the total path between these two leaves, and the distance between this and this is the length of the total path here. But if I know for any two leaves this distance, I get a distance table, that is a perfect encoding of the tree structure. I get the tree from this up to topology and even up to metric topology, so that even as a metric space can be reconstructed completely. We come to that later. From these degrees of resemblance, we try to derive a distance table, and from the distance table, we try to construct a tree. That's the general idea. And so, you better check your distance. And that's you read this on the highway in England all the time. Check your distance, and they're absolutely true. If you try to do phylogenetic analysis, check your distances. So, but why not just be happy with trees? Why phylogenetic networks? In phylogenetic analysis, such phylogenetic trees are, of course, the object of desire. That's what we want to see. How did it all evolve in various branching processes? However, taking phylogenetic networks into consideration too has a number of important advantages. They can be used as representing entangled or reticular evolution or hybridization events. Then you don't have a clear-cut family tree, a pedigree, a phylogenetic tree, and if you don't have that, you can represent that still in form of a network. Graphs whose universal cover contains a desired correct tree. If you say look at mitochondrial genomes, and most of the time you just look at the purine pyrimidine alphabet, so you have just a very simple two-letter alphabet, and so these are all points in something like 0, 1 to the n in a hypercube. So all these mitochondrial genomes can basically represent it as a point in a finite dimensional hypercube. And in this hypercube, you would connect two vertices if they are connected by an edge in the hypercube, for instance. If you have enough data, that's good enough. And then very often you get, we have, there. Very often you find sort of some vertex, the neighbor vertex, this vertex, and this vertex. If we would just have dimension 2, this could be 0, 0, this could be 0, 1, this could be 1, 0, this could be 1, 1. And maybe here are many of those, and here are only very few, here a little bit more. So what is the evolution of this process? Maybe it all started here because there are still so many of this, but then the second site mutated to 1, also the first site mutated to 1, and finally both sides mutated to 1. In which order did it go? Nobody can tell. Probably, if there was enough time for all of that, and these were highly mutable, actually it could have happened this way and this way. So in mathematical terms, you would look at the universal cover of the square. This is a graph. Universal cover is rather infinite, but it's a tree. And the given tree could be just the tree of evolution is in most cases just a subtree of this. We can absolutely be sure in mitochondrial evolution there isn't even, you get it all from your mother, your mitochondria. So there's no problem with crossing over events or whatever and mixing, sexual mixing. It is very clear-cut evolution. There must be a tree. I'm absolutely convinced of that. But if we see this, the tree is that we really want to see is somewhere in the universal cover of the square in mathematical terms. It could also be, you will see later a few images, just superpositions of trees can be visualized in a network. For instance, when Alan Wilson, New Zealander, who they still have the Alan Wilson Center now, but went to America, and he published this famous paper about the African Eve, where he claimed, together with some co-workers, that after investigating the variety of mitochondrial human sequences, that the Homo sapiens must have appeared somewhere in Central Africa, something like 160 to 180,000 years ago, and then began to spread first all over Africa and then all over the rest of the world. About 80,000 years ago, they went into the Arabic Peninsula, and from there they spread west and east. The people who spread west were probably then the forefathers of the Basques. The Indo-Germanic tribes came much later into Europe, and the other people who went east populated Asia. Some even went down to Australia, where they arrived 50,000 years ago already. Some went over to China, some went into Inner Asia, and some from there over to America. So there was this continuous spread, but that was about 80,000 years after Homo sapiens originated, by what we don't know. And this means that in Africa there are many, many more variants of humans, at least on the mitochondrial genome level, than in the rest of the world. They are all together. That is in the phylogenetic tree. If you look at the tree, you would see Africans all over the tree. In each branch there would be Africans. Europeans only in one or two branches. Asian people, Australian people only in one or two branches. And a few. This was a very impressive paper, and almost everybody now believes this is true. However, people were soon to point out that the one tree that was used by Alan Wilson and co-workers was only one of many almost equally good. So he used a sort of most parsimonious tree, and given the data, there are hundreds, if not thousands, of equally parsimonious trees. So how could you base such a far-reaching conclusion on just one tree where you have thousands? Now, other New Zealanders soon came to help, like Mike Handy and David Penny, and they just investigated not several thousand, but I think a few hundred of these most parsimonious trees, and they all showed the same phenomenon. The Africans were all over the tree, the Europeans and the Asians were only at certain branches. So that sort of corroborated this conclusion by Alan Wilson. But maybe if you can put all these trees sort of into one big network that is sort of like a superposition of all of these trees, and then you see Europeans only at certain places, Asians only at very specific places, and African all over, then you don't have to look at 100 and more 1,000 trees altogether. But most importantly, networks can represent our huge ignorance. Really, all that happened a long time before I was born and before you were born. And so how in the world can we really know how insects evolved, how mammals evolved, how whales evolved, and so on? It's all guesswork. And much of that is really, you very often you have sort of, gorilla, chimp, and man. Okay, in former times, of course, we all believe gorilla and chimp are close relatives because they all have fur and they all live in Central Africa, not so far from each other. And humans must have separated much earlier. Then from genetic data, another picture emerged. Apparently, the ancestors of chimp and humans separated from the ancestors of current-day gorillas about maybe 8 million years ago, and then only chimp and man separated maybe 6.5 million years ago. And this can amazingly be correlated with a geological phenomenon. You know, the great African Rift Valley that came about 6 million years ago, plus minus, it's always rough estimates. And you find chimps only on the western side in the Central African rainforest, and you find remains of humans, hominids, only on the eastern side. So apparently, humans derived from all those ancestors, sort of the pre-human chimp people, who were on the eastern side of the Grand Rift Line. The other were on the western side. On the western side, the Rift Valley, on the western side of the Rift Valley, it remained wet enough to support rainforest. On the eastern side, it became drier and drier and drier. So whether we wanted it or not, there were not enough trees to climb around. So we had to come down from the trees. We had to survive in the savanna and develop, or we could have as well decided to die out, but we didn't. We somehow developed certain abilities to survive. I guess what we actually used was something we brought down from the trees, because we were weaker than the animals we could have eaten. We were even much weaker and not that fast as the animals that would like to eat us. And it was not so obvious how to survive. However, we had a very good optomotorical system. That was based on our visual cortex. That is, climbing and jumping from branch to branch in a tree, you have to look at the tree you want to grab, but then you jump, and while you jump, you have already to look at the next branch, and still you have to know where the branch you want to clinch to is. So that is all being processed in your brain. You think you know exactly where the brain is, but you don't, where the branch is, but you don't see it anymore. It's all in front of your inner eye. And I think this ability to make very good visual representations of the outside world that are sufficient to work first on the visual representation before you do anything. This enabled us to produce tools. To talk about, to communicate about these visual representations, these virtual representations of the world, we needed language. So we really needed another language system than the communication system even of the chimps. We had to develop it anew, so I'm very happy with Chomsky in this context. He says it's really something else, the human system. It is apparently evolved to communicate about what is before your inner eyes, and so on. So that happened in the last 6 million years on the western side, on the eastern side of the Great African Rift Valley, and that what finally brought about the humans, I would think. But let's go on. I hope we can. Yeah. It's here already, but now it must. There it is. So, in principle, but now coming back to humans, chimps, and gorillas, now we believe that really the closest relatives of the chimps are us and not the gorillas. But who really knows? So it's always, current data say this, but really the ignorance about what really happened is still amazingly huge, and therefore maybe it would be better to represent the whole thing in terms of superposition of the one tree that connects the man with chimp and the other tree that connects chimp with gorilla. Furthermore, it is not even clear that the object we want to investigate evolved in a tree-like fashion, if it is not clear. And if, first of all, we want to find out whether this was the case or not, trying to anyway represent Darwin's degrees of resemblance in a tree-like fashion will not help to answer this question at all. The case in point being the necessarily prebiotic evolution of the family of transfer RNA molecules. You know, before life can start, we need transfer RNA molecules. Or the first cell, the first living cell had to have transfer RNA molecules. But not just one, but one for every amino acid that was being used in the proteins the cell was trying to construct. And they are all very similar, so they must have evolved in the primordial soup without Darwinistic principles, first of all, but maybe by Darwinistic principle. If you have replication, mutation and selection on the molecular level, as Manfred Eigen pointed out, then you can even have Darwinian selection processes, Darwinian evolution in the primordial soup. And Manfred Eigen believed that the transfer RNA molecules came about by such a process and then were incorporated into the first living cell when sort of encapsulating all these interacting molecules into one big membrane became the fashion of the day. So that's actually what also brought me into this topic. Manfred Eigen, when he was trying to exactly investigate the evolution of transfer RNA molecule, at that point you couldn't go to Google and look for a good software to produce a tree for the transfer RNA. What he could do was looking at Margaret Odehoff's Atlas of Proteins, where she also dealt with transfer RNA, and she had also trees. And then he wanted to understand how did she come up with these trees, and he realized it isn't that easy. It's not so obvious. Just because of such phenomena. If you have something here and here and here and here, and you want a tree, you have to cut one of these edges. And how do you do this? And then he had noticed in one other conference that me being a mathematician was interested to get into some contact with biologists, just to learn some biology, because I was terribly ignorant. And so he invited me to come over to Göttingen, explained me his problem, and then I began to think about that and try to find algorithms that could construct a tree of transfer RNA, but first of all could answer whether the family of transfer RNA molecules really came about by evolution, by prebiotic evolution, or whether these were just all very similar molecules. There were many molecules like that swimming around in the primordial soup, and when the first cell came together, it just selected a few of those. That's the alternative. So that's similar. They are all similar. Are they similar because they are similar function and were selected because they had that function, or are they all similar because they came about from one primordial first transfer RNA? This we had to answer first, and so even if there would have been neighbor joining as a very good method for reconstructing phylogenetic trees, we couldn't have better used it because that presupposes that there is a tree. But we wanted to know, was there a tree? And then neighbor joining, of course, can't help. Actually, creationists, once again, noticed very well then that all the trees you now find in all publications on this matter are constructed by algorithms that presuppose that there was a tree. And so therefore they say, oh, we can't believe, we don't need to believe, and we can't believe in these trees, because if you presuppose that there's a tree and you find a tree, that's not a proof. And so we wanted to really construct an algorithm, construct a method by which we would sort of understand phylogenetic family relationships between sequences without presupposing that the basic relationship between these things must be visualized by a tree. Being remarkable, constructing networks from a table of observed degrees of resemblance turned out to be much easier than expected. So when I began working on that, I had no idea, and there was no other literature that I knew about. There was some, but I didn't know about it, that I could have referred to. But it's very easy. Given such a table with one entry, dxy, for any two species xy, that is the distance of x and y, not the similarity, but the dissimilarity. One just has to consider the convex polytope of all maps from x into R. So these are elements in R to the x in a linear real space, such that fx plus fy is larger or equal to the distance between x and y. And this is a convex polytope, which is of course non-compact, because if you have such a map, you can always make it larger. It will still satisfy this inequality, because there's nothing that bounds f from above. But now you take the union of its compact faces. And this is always a contractible compact metric space. And in case the observed degrees here of the resemblance fit exactly into a tree. Say if you had exactly the length of these passes in Haeckel's tree as your input matrix, this union of the compact faces of this funny polytope would be exactly the tree. So this is how you can reconstruct a tree from the distances between the leaves. Of course, there are other ways to do this, but this is for me the most conceptual way of doing it. But if it is not, if this does not fit exactly into a tree, then you won't find a tree, but you always find at least a contractible compact metric space. It's just higher dimensional. And it's actually not only a compact space, it's a cell complex. And so it has vertices, edges, and higher dimensional faces. And so this cell complex maybe is a good substitute, I thought, for the phylogenetic tree. Sometimes now higher dimensional convex polytopes or just the compact boundaries of higher dimensional convex polytopes is probably difficult to imagine and even more difficult to sell to biologists. So together with Daniel Jusen and who was sitting here and many other friends and students at that time, we tried to develop methods, in particular Hans-Jürgen Bandelt needs to be mentioned here. We try to set up ways to extract information about this union, this T of D and P of D, that we could visualize and then come up with networks. And furthermore, as degrees of resemblance can be observed from many beings, not just for species only, we can test procedures for investigating phylogenetic relationships, H is missing, on the basis of degrees of resemblance from many classes of objects. So we can test procedures for investigating phylogenetic relationships on the basis of degrees of resemblance for, yeah, that's, okay. There's something wrong, but anyway, I wanted to come to this. This we could delete. Colors, manuscripts, towns, languages, and so on, not just species. And that's what we will do now. The phylogeny of colors. Of course, it's not a phylogenetic tree. It's clearly a network. Some guy, Helms, got his PhD in around 1980 in Hamburg, and that was a paper that was discovered by Hans-Jürgen Bandelt, whom I just mentioned, where he tried to measure the perceived color similarity of colors. So he had many test persons, and they always, one color was here, one color was here, and they got a third color, and so they had to put it somewhere between those two so that similarity relations between the colors as they perceived it were sort of corresponding to the distances between these three color papers or whatever, clips. And then he measured from the data he came up with some kind of distance, dissimilarity, how dissimilar do I find certain colors are. And if we used, so this is sort of a two-dimensional projection of parts of this convex polytope, T of D, or the boundary of this convex polytope. And you see it fits very well. You get a color circle. Everything is very nice. And you see there is some signal here that separates the bluish, reddish colors from the greenish, orange colors. There's one signal here that satisfies the greenish, bluish from the orange, reddish colors, and so on. And the whole perception can be interpreted as the weighted sum of all these perceptions of distinct colors. So that, for instance, here, this separates these two. The intensity is not that large as the intensity here or the intensity here. There's quite a high intensity separating these two colors and so on. And if you now look at the total distance between any two colors here along the shortest path in this network, then up to 1 or 2%, that is exactly the measured distance by Helms. So this is quite amazing. We were very happy. Now Helms also looked at colorblind people, and in this case we get indeed a tree. But the correspondence is not 99%, it's only 70%. So this is apparently red-green blind people. There's no split that separates the reddish colors from the greenish colors. But they can separate very well these colors. So clearly they should not get a driver's license because all these colors here, yellow, green, and red, are close together. we also looked at a road atlas. We took a distance table of German towns, Cuxhaven, Bielefeld, where I lived for so long, Berlin, where I was born, Basel, Bayreuth, and so on. And then we used this algorithm to produce this sort of projection of T of x. And okay, Cuxhaven doesn't necessarily to be in the north, and east and west might also be just by, replaced by mirror image, but up to putting Cuxhaven in the north and putting Berlin rather than Aachen to the east, we get a pretty nice representation of German geography that everybody who knows a little bit about Germany will know. Bayreuth, where the Festspiele are, is somewhere in northern Bavaria. Augsburg is in middle Bavaria. Basel is far in the south. Aachen, Bonn, North Rhine-Westphalia in the west, and so on. So it's a pretty good representation. This is even more interesting. This is diagram that has been constructed by Peter Robinson and co-workers now in Leicester, in Montfort University. Then he was still in Oxford, and together, when I was here the other time, I went with, were you there? No, but with Mike and with David. We went to visit him in Oxford. He was working on the manuscripts of Canterbury Tales. There are about 150 manuscripts of various parts of the Canterbury Tales, and scholars had studied their life long to classify these things, to find the most original version, and so on. After all, at that, he was, from European point of view, he was 100 years too early, because no book printing had been available. So if somebody wanted to have a copy of Canterbury Tales, this is running out? Not quite, okay. If somebody wanted a copy of Canterbury Tales, he had to send a scribe to copy the book at somebody other's place. That's how it was. And there were more than 100 such copies around. Now, Peter Robinson just used 58 manuscripts, and he looked the wife of the Bath, the prologue. It's only a few hundred letters in the text, maybe 800 or so, and he counted differences. So again, he came up with a distance table. Of course, just a simple spelling mistake was not a distance, but replacing one word by another one was, that was really serious. And if you find the same words then in many texts and not in those texts, then clearly you would better separate these texts from the others. So, but now he only looked at the distances from counting all these serious changes, and he found this diagram, which is almost a tree after all. But it's not quite a tree. Here's some non-tree likeness, and here's some non-tree likeness. It puts all the manuscripts which scholars that had worked all their life long on these manuscripts into group CD over here. Group O is over here. That is rather, sort of rather close to the origin. This is Hengesworth's manuscript. I think that's the one that you can still easily see if you go to Huntington Gardens, where this is a marvelous library, and one of their pieces is a Canterbury Tales manuscript, and that's one, so that's almost, must be close to the root. Here group B, group E, and so on. They all come out very nicely with this thing. So this that this distance that Peter Robinson constructed from just looking at the manuscripts comes up with a relatively good classification of these manuscripts. And again, you don't need anything else but just looking at these networks, and that this network was close to a tree was fine, but for Robinson it was very important to corroborate his idea that there was a tree-like development for the manuscripts. If he could have, of course, used just neighbor joining and come up with a tree, and that would have been the tree, but then there would have been a discussion, does that really fit to the data we are looking at? Now with 16S rRNA, ribosomal RNA, we come up almost with a tree. So for me, this is the best argument against creationism. Because it could have been a network as with the colors, but it really is a tree. There are some non-tree likeness, but that's very, very small. It also supports the view of Woese that all the archaea, there are 3 big groups of archaea, but they're all clearly separated by a little split from the eubacteria, including the chloroplasts and the eukaryotes. So we also see that the chloroplasts that you find in tobacco and in corn really came along by a symbiosis between plants, or between bacteria actually, maybe green algae, and some bacteria, between plants and bacteria, because these are really bacterial RNA molecules. And they are very close to Anocystis nidulans, which is just a bacterium. Has nothing to do with plants and photosynthesis. And you see here, sort of, we would be very close to mouse, almost indistinguishable. So on this scale, mouse and man are almost identical. Even Xenopus, the frog, is close by. And even here, yeasts are still on our side in this thing. But you can, so this was very long-term evolution. You can also use it to analyze very short-term evolution, sort of the AIDS virus. Here's the AIDS virus, and you always look at the first position of coding genes. You know that according to the genetic code, there is redundance. The first position is rather conservative, because if you change the first position of a code word, you get, you might always, not always, but you might very easily get another protein, amino acid being encoded by this triple. But the first position doesn't change much, and as HIV changes so quickly, you have to use methods that focus on what changes slowly. So the first position in coding genes, slowly. So you have HIV1, HIV2, mandrill, chimp, African green monkey, macaque, and so on. And what you easily see is that there must have been 2 independent infection events. And according to people nowadays, I mean, you can think of anything, how chimps come so close to HIV1, but it's just likely that people ate chimps. And that this way infected themselves, and that was how HIV1 came about. And this is also in the place where chimps are being hunted and eaten, while they may have eaten smaller monkeys. But you can now, okay, this depends very much on your distance. Check your distance. What can you do? You can just use the same data, but you can try to interpret them in another way. Try to construct another distance on the same data. Doesn't want to give way. Here we just took the whole sequence, but we did not take into account the difference between 2 different purines or 2 different pyrimidines. We only looked at the sequences in the RY alphabet. And then you basically, but otherwise the same data, you basically get the same structure. Again, it's almost a tree, again corroborating the idea that there was a sequence evolution. Chimp is close here, mandrill is here. And it's also interesting that here you really see some kind of tree-like evolution going on. Here, this is like explosion. This is all from one patient, and this is from different patients. But it's typical for evolution of sequences where basically everything goes. That is, if you look not at HIV, but at the influenza virus, you would never see such a picture. The influenza virus has to fight with our immune system, so our immune system develops some trick to eliminate influenza virus. Then but a new variant survives and will again fight with our immune system. Then our immune system has to learn. So there's really a very well-controlled evolution going on between influenza virus, and that leads to perfect trees. Tree-like development has been investigated very thoroughly in the last 10 years by Walter Fitch. But HIV, everything goes. It doesn't have to fight with the immune system because it makes itself part of the immune cells. And that's why it so far has been able to evade our abilities to fight with people that with viruses and so on. And that's what I told you about already. This is this network that is really from Daniel, network of human mitochondrial DNA. You really see, first of all, it's a network. So we are absolutely convinced that the evolution of mitochondrial DNA was tree-like. So it's rather sort of the real tree must be somewhere in the unramified covering of this graph. But what you see anyway is that Europeans are here and here, and that here's one more, but they're all over here, and also Asians are here on this side. So probably somewhere here, these dots represent those little bands of people who came out of Africa in 80,000 years ago and went into the Near East and then spread over the world. They're still related to all sorts of Africans, Yorubans, Hereros, African Americans, Yoruban. But here are also, these are all Africans. African Americans, but that doesn't matter. Here's one Asian, so it's not so clear how this comes about, but okay, first of all, there can be individuals who can have just come with a group who had another setup, genetic setup. But I think for me, this is the best way to corroborate Alan Wilson's hypothesis that we came out of Africa. And you can apply it to languages. You see, and here we use the neighbor net construction on distances that were derived from WALS, W-A-L-S, the World Atlas of Language Structure. Because as Tandy, is she here? No, but she gives a lecture tomorrow. Will explain to you, if you want to construct a tree of languages, you better restrict, say, to Indo-European languages, where you can compare words. Because we basically have the same corpus of words, and then we can see how words have been changed in Greek and French and so on. Now in Leipzig, in the Institute for Evolutionary Anthropology, they constructed this World Atlas of Language Structures, where for about 5,000 identifiable languages, they asked experts to fill out forms that would specify 138 character values for 138 characters for these languages if known. So apparently this matrix is not completely, or this table is not very filled up. There are many languages where we just know too little, so to do any kind of numerical analysis. But for these languages, these are of course not 5,000, but only maybe 25 or so, we do know enough to make a numerical comparison between the various character states of the various structures. Structure means, does a verb come in the middle, at the end? Does the adjective come before the noun or after the noun? And so on, all these things. And you see German and French clearly are close related. Modern Greek is not so far. Then comes Hungarian, and if you would have let Finnic language would also be here. Hebrew, modern Hebrew. So this comes all together, which sort of explains that Semitic people, Hungarians, Finnic-Ugric people, and Indo-Germanic people, they all came from Central Asia with the Neolithic Revolution. the Western world and are related. The Basques, other Europeans, are far down here, closer to Georgians and people where we do not even know what the name of the language is. Mandarin, Korean, Japanese is all closely together. So it's quite interesting. We don't see that this is really language evolution. What that represents is sort of similarity relations based on language structure. And language structure does not really mean that one language comes from the other, but certain tricks of putting your words together. They easily go from one place to the next. So now my thanks go to you for your attention and patience, the INN and the organizers of the Phylogenetics program for allowing me to participate in this most fascinating and inspiring event, to Daniel Lewson and Mihai Albu for generously providing me with their JPG and PPT files, William Edgley for introducing me into the martial arts of fighting against, not with, Bill Gates' PowerPoint program, and to my present home institutions, the PICB in Shanghai and the MPI for mathematics in the sciences in Leipzig. And that's it. But I think I still have a few minutes. Okay, a few minutes, so I can, maybe we can get this up and everything stopped. Yeah, that comes help. So how does it all work? How do we get these diagrams? I mean, these diagrams are nice to look at and they seem to tell interesting stories, but how do we get these diagrams? This is amazingly simple. What you really want in biology to start with, you do not, okay, you want something like that, but more precisely, you want to have splits among the species you investigate. So you want to have the split that defines the birds and separates them from all non-birds. You want to have a split that separates the mammals from all non-mammals. You want to have a split that separates the vertebrates from all non-vertebrates and so on. So always, if this is the set of species you want to investigate, you want to have splits. You split it into all possible ways. Now, if these splits are phylogenetically compatible, that is with birds and mammals, you would indeed have maybe birds and non-birds. And here, mammals and non-mammals. Clearly, no mammal is a bird, so this is intersection. The non-birds include the mammals and many things others. The non-mammals include the birds and many others. But you do not have a bird that is a mammal, except in mythology, but not really. But if you would say flying and non-flying, okay, you can have something here and something here and here. I mean, not all birds fly. Well, basically, birds fly, but not all of them. Basically, mammals don't fly, but some still do. So you have something everywhere. And so you know, of course, phylogenetically, this is not a good split. But for other reasons, it might be a good split whatsoever. And so in phylogenetic analysis, we are really interested in given our set to find all ways of cutting it up into splits. Moreover, any such split, I mean, think of this is a metric space, so here are points that have their own distance, not the 2D distance you see. Giving such a split, you can associate a weight to it. You can say, yeah, it's a strong split. So the split between mammals and non-mammals is a pretty good split. because mammals separated from the non-mammals something like 300 years ago. 300 million. It's always in millions. Maybe 280 or so. So that's a pretty good split. But the split that separates cows from horses is not such a good split, or so on. So you would give, you have a split. You first have a set X, then you have the set of all splits of X. So it contains just bipartitions of X, A, B are contained in X, and their union is all of X and the intersection is empty. And we have something like a weighting. And we have this weighting really. To each split, we associate a non-negative real number. Some of these numbers may be 0. That means this split doesn't seem a good split from biological things. Others may be positive. So we have such a weighted family of splits. And these diagrams do nothing but representing this weighted family of splits. I mean, you saw these splits. If you think of the colors, you saw these parallel lines. If you look at the sequence of parallel lines, there's something to the left, something to the right. And one can, on the one hand, one can look at these diagrams as you saw them, the images. From a more formal point of view, they form a category. I call these diagrams X-nets, and there's a category of X-nets, and in this category you have morphisms and all you want. Actually, it all started with Isbell observing that the metric spaces, it was 1897 or so that he published this paper, that the metric spaces form a category. Okay, the objects are the metric spaces. What are the morphisms? Continuous maps, too many. Isometries, isometric embeddings, too few. The right thing is non-expansive maps. So you can map one set into the other, but 2 points that are far apart here should not get further apart here. They should either get together or have the same distance. With this category of metric spaces, Isbell showed that every metric space has a unique injective hull. And if the injective space is X is D, and D is such a distinct table as we defined it before, TD, together with the induced L infinity metric, is the injective hull. So quite amazing that concepts from category theory in this context make sense. And now you can go on working on these things, and you can make similarly a category of X-nets, where you have vertices and edges, and the edges have certain lengths that induce a certain metrics, and you can define what are good maps. And again, non-expansiveness and so on is important. And again, every X-net has an injective hull, which is sort of the X-net with as many edges as you can put in. And the injective hulls are exactly, or the injective X-nets correspond to these weighting systems. Just that. Very simple. And the art, of course, of drawing these maps is not to drawing this injective hull that has much too many edges, but trying to forget as many as you can. So Daniel spent a lot of time to find the best way to drop edges that we do not really need. But there is a conceptual framework for these images that you saw. From my point of view, these images sort of represent X-nets that are basically characterized by such splits. I mean, after all, you saw the splits. Only going back and forth and so on needs some machinery to be developed. But what I wanted to point out here was that it is not just nice images that tell nice stories, but it is also, there's a very rigid mathematical structure behind all that. And without that, I think we wouldn't have been able to develop all of these nice programs. You find all these things in Splitstree incorporated, and using this software, you can construct as many of such nice images as you want, provided you have the data. And but the other, and what what Splitstree really does, if you input, you can input other things, but if you input distances, it doesn't compute this, but it computes a projection of that and looks at the splits that are supported by the thing and draws then the XNet given to this set of splits. I think now my time is over, so thanks again for your attention, and we can have a discussion with you.
Thank you very much. So I'm sure the discussion is between us and a glass of wine, but we should have a discussion. Questions or comments?
Can't we have both?
And both, yes.
Simultaneously.
Yes, please.
Andreas, in your list of 5 reasons to use networks, I think there was one that there's one reason that you might want to add, something that Dave Bryant came up with. If you think about this statement, the average German family is 1.3 kids. There's no family in the world that has 1.3 kids. But as a summary statistic, that's much more precise than saying we have to restrict ourselves to whole integers for numbers of kids, because you can either say average family has 1 or 2 kids.
Yeah.
And so here, the truth might lie between, a network might be a better way to represent, a summary statistic than a tree.
Another good reason, yes. That's why I said it's a sort of a superposition. This tree might be representing the history of the first half of your chromosomes, and this might be representing of your second half, but you get it all together, whatever.
I had a rather a naive question. I think of a tree as really a very special limit of the kind of more general nets that you were drawing. So I can't help thinking you only got through that by actually excising things with a certain cutoff, as it were.
Yeah,
is that
or
the way I think about
contracting.
it? Or contracting, yes, through a certain rule. So would the creationist get at you by saying, well, you're just doing the same through your contractions, as it were?
Now, if the networks I get are so close to a tree, they are just a tree plus epsilon, then I can easily refuse this argument. Because I contract something in this image with the ribosomal RNA, 16S RNA. Actually, I increased the length of the edges that were skewed to the tree-like structure, just so that you would see it. Otherwise, it would just look like a tree.
Right. Good. Any other questions? Yes, please.
Looking at your diagram on the right there, in the, I don't know anything about biology or maths, but the going from 0, 0 to 1, 1, if that involves switching 2 bases, then it presumably can happen quite often at either order.
Yeah. That's what I mean. It can actually go this way and it can actually go this way, and it could have gone both ways. So that's why I say you go to the universal covering of the whole thing. There you have it all. So you would sort of try, if you see this, you would, and if you could find out that there was some ancestor with this, so it came this way, and if you see it at another time and you know there was some ancestor that was this, so in the universal covering, together with the ancestral history, these would be 2 different points. That's exactly how you construct the universal cover of any space. You have one base point, and then you look at all possible paths from the base point to the point in question. And if there are non-homotopic paths, okay, then there are different evolutionary routes for what has been developing. So I think this is not just a metaphor to talk here about universal cover, but it really means something. And they are not only the human mitochondrial data. A guy from New Zealand has been going down to Antarctica again and again to collect frozen penguin remains from up to 30,000 years ago, and he has been sequencing their mitochondrial RNA to better understand the evolution of mitochondrial RNA, now in particular in penguins, because there we can really go back 30,000 years. And he comes up just with the same thing. So again, many types he finds again and again and again, sometimes are rarer, but his diagrams are full of these situations. And so in 30,000 years, of course, these penguins had time to go in various directions and sort of come to the same place in different ways.
But not just naively a tree.
Something you may not know, although Linnaeus created this hierarchical system, he also changed his ideas on the species concept. He was actually the first to produce an artificial hybrid, and he came to think that hybridization was actually very important in plant evolution. So that was quite late, and it's not often mentioned actually, because people think of him always in terms of this hierarchical system of King Philip.
Who was this?
Linnaeus.
Linnaeus
Yeah.
already.
Yeah, he was already thinking about this. He had some crazy ideas of some plants that he thought they were hybrids. They were not, it turned out. But he did make the first artificial cross of Tragopogon species to produce.
He was in charge of a botanical garden anyway, and there was hybridization going on.
Yeah,
and
that's
people
true.
even tried it, yes.
Yeah.
But of course, you know much more. I mean, I'm so happy that he is here, because he is a down-to-earth biologist, plant biologist, and he is using all these network construction methods to better understand the relationship between buttercups or whatever family of plants he's investigating. So Pete Lockhart from New Zealand, for those who don't know him. And if you don't believe me that networks are good in biology, you should ask him.
Or even beyond networks. Yeah, networks beyond trees. Great. Well, maybe that's a nice note to finish. People we can talk informally afterwards. I think this has been a fantastic example of a Rothschild lecture, actually, because it's been so accessible. Really grateful for the effort that you made to put that together to make it so accessible. Fantastic breadth. If you look at what we've covered, apart from these wonderful historical insights that we've had through Chomsky and other language analysis of different forms of HIV, etc. And I think taking us as well beyond the rather naive picture that I had of phylogenetics as just being associated with trees. So therefore, for me, certainly this has been a revelation. It has made me absolutely determined that I've got to have a little library of examples from your guys' program, which I can tell other people about the Newton Institute.
Daniel can.
So that's my little program for the next 2 or 3 weeks. So should we all join, please, in thanking Andreas for a really nice.
Thank you very much. Thank you.
And everybody's now welcome.